Good afternoon, everybody. Wednesday afternoon, and it's lockdown learning number 47. So welcome back, and I hope everything's going well. So let's move on. So let's have a little think about what we're going to do today. So I managed to come out of number 46 without revealing anything about what we're going to be talking about today, because at the time I frankly had no idea. Obviously since then I've been doing a little bit of thinking, and I've got some ideas for the next, I think probably the next couple of lessons. So definitely for today, and whatever we don't get through today, I will shift over into Friday. Number 48. So a little bit of a quick recap of the last little while. So since we came back from our break, we've had a look in a reasonable amount of detail at short read alignment. So taking a high throughput sequencing experiment, typically from an Illumina sequencer, aligning the reads in that experiment against a reference genome. So for our demonstration purposes, our reads came from a fairly randomly chosen genome sequencing experiment on the sequence read archive. So we looked at an experiment where they'd sequenced an E. coli genome, and then we aligned those reads to the E. coli K12 reference genome from GenBank, which was seemed to be a reasonable thing to do. So we had about 1.4 million paired end reads in our sequencing experiment, aligning against the 4.5 million roughly base pair E. coli reference genome. So that we've done all of that, generated our alignments, and we've talked then reasonably in again a reasonable amount of detail about those alignments. So about the SAM format for storing alignments, and what's encoded in a SAM file and how it's encoded. We also talked about last time out we introduced SAM tools, which is extremely well established, well developed software for working with and manipulating SAM files. We particularly looked at compressing our SAM files, so going from SAM to BAM using SAM tools View. But we also looked at sorting our SAM files in coordinate order and creating an index for that sorted SAM file as well. Sorted BAM file as well. So that's that's been the last little while, couple of weeks, I guess. So what I'm going to do today is talk a little bit about some things that we can do with that BAM file. So today and Friday is going to be about kind of things that we're going to do with that alignment now that we've generated it. Particularly today, I'm going to look at visualizing alignments. So we're going to look at a tool, a widely used tool for visualizing the alignment of short reads to a reference genome. So we'll look at a genome browser, and I suspect that will take up quite a lot of this hour. If we have time, I'll move on to something else, but I think that I'll probably be able to prattle on about IGV for an hour or so. So yes, genome browsers. So a genome browser is a piece of software that is used for visualizing genomic intervals. So you can look at anything which you can consider in the context of its genomic location, you can visualize on a genome browser. So The most sort of common and popular genome browsers that you may well have come across tend to be online, so tend to be web sort of web applications. Particularly, you may have come across. There we go. Get rid of that. The UCSC Genome browser. And these have largely been developed in the context of the human genome, although they do provide other genomes for browsing as well. But you may well have come across the UCSC Genome browser, which is extremely popular, particularly has kind of been developed alongside the human reference genome, so is particularly popular for viewing the human reference genome. So here we go. This is GRCH38 reference genome. We're looking at, in this case, a small portion of chromosome X. So this is actually the ACE2 gene. So here we go. Here is this little red bar that you can see here. That's the bit of the genome that we're looking at here. And then in the square window down at the bottom, we can see the so-called tracks. These are the visualizations of the features that are present in that particular little window of the genome. So you can see at the top we have the gene tracks. So we've got these are the exon and intron structure for the ACE2 gene, which you can see is running. The little arrows tell me it's running from right to left, so on the negative strand in this particular region. There's some gene expression data for that particular gene here as well, histone mark data, DNA's hypersensitivity assay, so DNA accessibility assay, as well as the conservation of this particular region of the genome between humans and up to 100 other vertebrate organisms. So this is the Philo-P score for this particular region of the genome. So you can see that in the exons we're seeing good conservation, and in between the exons not so much. And there's specifically alignments here between the human genome and other key vertebrates here, so monkey, rhesus monkey, mouse, dog, elephant, chicken, etc. And then some information about single nucleotide polymorphisms taken from dbSNP, and repeat elements in this region of the genome as well, from repeat masker. So you can see that even in this relatively small window fits in one go in my video window here. You can see quite a lot of information about this particular region of the human genome. Another example of a genome browser, one that again that we've already sort of come across already, is Ensembl. So Ensembl, which we have come across in the context of annotation. So we saw this website when we were talking about Biomark particularly. But Ensembl is also a genome browser. Also has a genome browser interface. So we can search, if we search Ensembl for the ACE2 gene, then we get the information about the gene, but we also get a browser, a genome browser version view of that information as well, and we can open the full Ensembl genome browser by going to view that region in detail. And again, Ensembl is showing us a lot of data about this region of the genome. So as well as showing us the ACE2 gene, it's showing us information about regulation in that region and various other things besides. So these are two web apps. They're websites which provide this kind of interface to reference genome data, and they're very useful and they're very good at that and very useful because of that. And particularly if you're working with particularly human genomes, but also other organisms which have a well-established reference genome, these tools can be hugely useful. Where they are potentially slightly limited in their use is a couple of things. So where you don't necessarily have a really well-established reference genome, so you don't that view of your genome of interest is not necessarily presented by default in these genome browsers. Or in a situation where you perhaps have relatively large amounts of data that you want to view alongside your reference genome of interest. And that is the real kind of USP of a standalone non-web based or non-web site based genome browser. Is I have a lot of data on my local disk that I want to visualize in its genomic context, and I don't necessarily want to have to upload all of that data to some web accessible location so that I can look at it in the UCSC genome browser or in Ensembl. I want to be able to view it directly off of my local disk, basically. And this is where a standalone genome browser will really come into its own. And the standalone genome browser that I typically use and recommend is the Broad Institute's Integrative Genomics Viewer, or IGV. So IGV is a piece of software that's been around for quite some time. It's been well developed. It's still actively developed. And this is a genome browser that you can download and run on your machine. And therefore, it will have access to your local disk. So if you have a lot of data that you've downloaded and you've got stored, you can load that data into this genome browser locally without having to make your data web accessible or anything like that. So we're going to have a look at the alignment that we produced last week, and we manipulated earlier this week to get it into a sorted BAM file. We're gonna take a look at that alignment alongside the relevant reference genome in IGV during this session. So that's kind of the goal of what we're going to do over the next 45 minutes or so. And so the first thing that we need to do is we need to install IGV. So IGV is a little bit like JalView, which we already looked at some weeks ago now. But JalView was developed as a Java application that you could download and run locally, but because it's Java, it's relatively portable, so it really wasn't too tricky to get downloaded and installed. And IGV is very similar to that. It's again a Java-based application that's relatively portable and is easy to kind of get installed and get running on our local system. So we are going to download the IGV desktop application and run that. And I'm going to do that on my laptop, on my host operating system, so on Mac OS. I'm not going to download this in my VM and run it on my VM. So much like we did with JalView, where we generated the alignment using command line tools on the VM, but then we viewed the alignment using JalView on our host operating system. Although actually, I think we did also use JalView on the VM. But in this case, I'm just going to go straight to installing it on my host, so on my Mac. So I'm going to download the IGV desktop application. So at the IGV website, which I found simply by Googling for IGV. So for me, at least logged in on Google, if I search for IGV, then the thing that I want is the top hit, and that I suspect is partly because of the things that Google knows about me. I don't know if IGV is a meaningful acronym for anything else in any other field, but certainly all of my front page seems to be about the Integrative Genomics Viewer. So hopefully the same is true for you as well. So you Google for IGV, then you should find it fairly straightforwardly. Otherwise, Integrative Genomics Viewer will definitely find it for you. So as I said, it's developed by a team at the Broad Institute in the States, and we can download IGV. So on the front page of the IGV website, there is an icon there on the left-hand side to download the IGV desktop application. So I'm going to click on that, and then, okay, I'm running a Mac. So I'm going to download the IGV Mac app and follow their instructions here. So it's talking about the fact that it's signed. They're a trusted Apple developer. They're signed as a trusted Apple developer. It's not notarized by Apple. So we need to do some special things to make sure that we run it correctly. There's also downloads here for Windows and for Linux as well. So whatever your host operating system, you should find an IGV download for you. So I'm going to download the Mac app. So it's version 2.8.3 is the one that I'm downloading. I think I do have IGV installed on my laptop already. I'm pretty sure the version I've got installed, which I ran this afternoon, was 2.5.3, so I'm a little bit behind. So it's good for me to catch up my my installation of IGV. So for Macs, it's downloading a .app bundle. So this is downloading a zip file, which I will unzip when it's downloaded, which just contains the app inside it. So in order to run that, I just have to move that app into my applications directory and run it from there. For Windows, it says it's to download and run an installer. So again, if you download that and run it and follow the instructions, it should install it for you on your Windows machine. So hopefully it's well developed enough software, and they're very used to supporting people that these installations should be relatively straightforward. So my download is 2/3 of the way there, 3/4 of the way there. So hopefully it won't take too much longer to download, and then we can go about making sure it's installed on our system, and we'll run IGV and take a look at how to use it. It is in lots of ways like a lot of point-and-click applications. It's relatively self-explanatory, and you can learn a lot about what IGV does just by exploring via the menus, the way that it works. But I think it's always worth, with these kind of applications, it's always worth taking a look with somebody who's used it before. So there we go. There's IGV 2.8.3. I'm just going to drop it into my applications directory. And then we can go there, and if I track it down, there you can see I've got an older version there which I've run before. We'll run IGV from there. Okay, so on my Mac, Mac OS is saying it's downloaded from the internet. Are you sure you want to run it? Which is just a security feature of the most recent Mac versions of Mac OS, just to be sure that you're certain that the applications that you're running are safe if they've been downloaded from various places. So I'm just going to click OK, and hopefully start IGV for me. So I get this little window when it starts. It says loading Genome. Just going to get this browser out of the way. Oh, I don't need VirtualBox right now either. There we go. So this for me is IGV starting up. So loading a reference genome in never seems to go very quickly for me on this machine for some reason. If you've just downloaded IGV for the first time, then the reference genome that it loads will probably be a human reference genome. I'm not entirely certain which genome is the default, but I have several reference genomes installed on my system because I use IGV for a selection of reference genomes installed already. Including the one which loaded for me by default here, which is actually, this is the E. coli genome that we're interested in. But I'm going to show you how to go about getting that, because you won't have that if this is the first time that you've run IGV. So I've got a selection of reference genomes installed. As I said, HG38 is kind of the standard, these days, the standard human reference genome, which may well be the default on lots of systems. So when the reference genome loads up, we can see here that we've got a relatively limited amount of information, but some information available to us. So we're seeing an overview of the whole genome here. So we've got the chromosomes listed along the top from 1 through to Y, with also some extra unassigned contigs here at the very far right hand end. And then we can see an overview of the genes in the genome, where the genes are in the genome. So if we zoom into particular locations, which we can do. So let's go to the X chromosome. We get an ideogram of the chromosome at the top, an overview which is showing us it's 154 megabases, sorry, and then here are again an overview of the genes on this particular chromosome. You can zoom into regions of the chromosome by dragging a window across the bar where it's showing you the size of the current region. If you drag across, so if we want to see the 10 megabases from 20 to 30, from megabase 20 to megabase 30 on this chromosome, we drag a box in that roughly in that window, and IGV will zoom into that region for us. So here we're seeing the roughly 10 megabases that I highlighted, and again we can zoom in. So if we want the kilobase here between 25,000 and 26,000, then let's zoom into that kilobase. So there you go, there's a kilobase of chromosome X, and the little red box up here at the top is getting smaller to show us which bit of the chromosome it is that we're looking at. So that is kind of generally navigating around. In an annotated reference genome, as loaded in as well, you can search for features. So we have genes loaded in with this reference genome as features. So we can search by gene name, and it will zoom us into that particular gene. So here again is the ACE2 gene, which we already know from those other two genome browsers that we were looking at earlier, is on chromosome X. So here is the ACE2 gene. You can see again it's going from right to left. This is the overall exon and intron structure of that gene, with the 5' and 3' UTRs shown as well by the little kind of slightly narrower region here. This is the 3' UTR of this gene. If you want to see alternative transcripts in the human genome, you can do that as well by right-clicking in the gene track, and instead of selecting the collapsed representation, if you select the expanded representation, then IGV will show you alternative isoforms for the genes in question. So in this case, with the expanded version, we see two isoforms of ACE2, which are essentially the same, other than in their 5' UTR by the looks of it. So just as a genome browser with some packaged annotation, IGV is sparse, especially compared to UCSC, the default view in UCSC, or in Ensembl as well. where there's a lot more data. But we can load data in to IGV to add to the richness. So if you wanted to load in some of the kinds of annotation that we were seeing in the tracks in the UCSC genome browser, we could absolutely do that, and we could load that data into view in this genome browser as well. But as I said, the versatility of this particular application is really in its ability to load in data that you have locally. So not these big publicly available data sets, but your local data sets that are individual to you. So particularly it's good for looking at alignments, for example. So that's where we're going to start this afternoon, is by looking at the alignment that we've generated for our short reads against our E. coli genome. That's what we're going to do. We're going to load that alignment in and have a look at that alignment and see the tools that IGV gives us for considering those alignments. So the first thing that we need to do then is we need to load the correct reference genome. So at the moment we're looking at the human HG38 release of the human reference genome. Clearly our E. coli reads have not aligned to this particular genome. So I've loaded already the E. coli genome, and see 000913 genome. But if you haven't, and you almost certainly won't do if you've only just installed IGV, well then it's more genomes are available. So via this, so in the drop down in the top left hand corner of the application, it allows you to select your reference genome, and the bottom option in that menu is always more, for you to pull up a list of the available reference genomes. So when you do that, when you click on more, IGV pops up this big list of available reference genomes. And in a lot of cases, it may well be that the reference genome that you're interested in is available in this list. So I can see lots of model organisms here, and lots of releases of genomes from lots of model organisms as well. So you've got 3 versions of the Arabidopsis genome. You've got a number of versions of the C. elegans genome, chicken, Drosophila, chicken, E. coli, which we'll come back to. There's a lot of versions of the human genome here as well, all the way back to B36, as well as HG19, HG38. Mitochondrial genome, you've got mouse, again, various versions of the mouse reference genome, and rat will be in here as well. Yep, so RN4, 5, and 6. Yeast, and various other things. We could keep going right down to zebrafish. We're obviously interested in E. coli in this case, because the E. coli K12 genome was the reference genome that we aligned to. And actually the reference genome that we aligned to, if you remember from our FASTA file that we downloaded from the NCBI, is this NC 000913.3. So we can check that actually. Let's grab our virtual machine. I'm going to have to restart my virtual machine because I upgraded VirtualBox today. So let's boot my virtual machine up. Booting. Shouldn't take very long. There we go. There's Ubuntu. I'm going to open my terminal, CD into share week, and this is our reference genome here. Sorry, this GCF file, that's our reference genome. So if we have a look at that, you can see from the header, it is NC_000913.3. Okay, so that is, as described before, that is this reference genome here. So E. coli K12 913.3, that is our reference genome. So we can match up the available reference genomes from IGV with the reference genome that we have used to align our reads to, which is quite nice. Nice to be able to do that. It's good that in this particular case we have used a reference genome which is absolutely a reference genome which is available in IGV. If you hadn't done that, so if you were using a reference genome that is not in this list, and although there's lots in this list, it's entirely possible that you will have used, especially if you're doing, if you're a bacteriologist and you're interested in a bacteria kind of outside of this fairly standard list of organisms, you can absolutely create your own genome in IGV. So you can create your own reference genome based on a genome FASTA file and some annotation. So you can load up your own genome should you choose to, as long as you've got a genome and maybe a GenBank file, something like that for annotation, for genes and so forth, then it should be relatively straightforward to create a genome file from your own particular reference genome. So you're not completely restricted to the genomes that are available through that list of genomes that we've just been looking at. Okay, so this is our E. coli reference, and we have, like we had for the human genome, we've got kind of an overview of the whole genome here. In theory, we've got some annotations. I can't really see the annotations at this point. Oh, there they are. They've appeared. So this is all of our genes, essentially, in our 4.5 megabase E. coli genome. No chromosomes in this case, or one chromosome in this case, because as we saw from the reference genome when we downloaded it, it's just the chromosomal DNA of the bacteria, so we don't need to concern ourselves with plasmids and so forth in this instance. So that's our reference genome, and as long as the reference genome, the names of the chromosomes in the reference genome and the name of the chromosomes that we've aligned to match up, then we can load in our alignment into here, and we will be able to visualize it. So we'll be able to visualize the alignments of our reads in our BAM file alongside the reference genome in this application. So this is what IGV is really good at. So that's what we're going to do next is load. So we're going to load from file. We're going to load our BAM file into here to visualize our alignment that we created. So I've picked load from file from the file menu, and then I've navigated to, in the resulting file browser that pops up, I've navigated to my week 10 directory on my shared drive with my virtual machine, and in there you should find, let's look at there's a list. There we go. You should find the BAM file here. So this is the sorted BAM file that we created in the last session, so in number 46. So last time out, we sorted our, among other things, we sorted our BAM file and we indexed it using SAMTools index. And you need those two things. You need the sorted BAM and you need the index for IGV to be able to load it in. So that is what I'm going to pick here. So I pick the BAM file, and IGV will load the BAM file, and it will also find the associated index, because the index has the same, the prefix of the name of the index is the same as the name of the BAM file, and it's in the same directory. IGV knows where to look for the index, but that index needs to be present for IGV to be able to load the BAM file. If I didn't have the index, IGV would complain and would then try to ask me if I wanted to create an index. IGV is able to create the BAM file index itself if you don't have one, but your BAM file does need to be sorted for it to be able to do that. So we're going to open the BAM file, and so this is now we have loaded the BAM file in to IGV, but because at the moment our overview is kind of the whole genome, so 4.6 megabases, that's too much, that's too big a region for IGV to be able to show us the detail of the alignment. To be able to see the detail in the alignment, we need to zoom in to a smaller region of the genome. We need to be looking at a smaller window to be able to see the details of the alignment. So at the moment, what it says in the tracks for the BAM file that we just loaded in, and it's created 2 tracks, what it says in those at the moment is zoom in to see coverage and zoom in to see alignments. So if we actually want to see the details in our BAM file, that's what we need to do. So let's zoom in. I'm going to press the plus arrow in the top right hand corner up here to be able to zoom in, and this will just zoom in on essentially on the center point of the window where we're currently visualizing. So it will zoom in right in the center of the reference genome in this case. So I'm just going to click the plus, and I'm going to click it a few times because we have to get quite a lot closer for IGV to be able to show us the alignments. So that's 3 or 4 times. Now it's having a think. I'm guessing it's going to try and load in our reads. So it's doing quite a big job because I'm still quite a long way out. So I'm still sort of 500,000. There's still 500,000 bases in my view here, and it's visualizing the alignments. I suspect that that's because I've got IGV set up so that it visualizes the alignments from quite a long way out. I think by default you'd probably have to get quite a lot closer in than this to be able to see any data. So that's not especially useful still. So I'm going to zoom in until it looks a little bit more useful. Okay, we're starting to be able to see a bit more detail. Let's go a couple more because it's nice to be able to see what's going on. Right, okay. So this is IGV. So with a BAM file loaded in, we get 2 tracks of data. So the first track, this top one here, is a track of coverage. So this is showing you an overview of how many reads are aligning to any particular region of this reference genome. So how tall is your tower of reads that's stacked up in the alignment that you're viewing below. So you can see that this ranges in this particular window. So this sizes dynamically. This ranges between from 0 down at the bottom to 167. So there's quite a range. Although actually mostly the coverage seems to be around about 140, 150 fold across this region. But you can see how it varies across this region. then the second track that's loaded is the alignment itself. So in this particular window here, each of the little blocks here is a read or set of overlapping reads. And so each of these grey blocks is a read that's aligned to our reference genome at that particular position. So you can kind of then see the arrangement of the reads against our reference genome, and you can see how well and how evenly covered this genome is in this locus anyway. It may not be across the whole genome. So this is a useful overview, but we can do more with IGV than just this. One thing to note at this very early stage is some information that IGV is already showing us just in this kind of default view, and that is that we can see positions at which the reads in our alignment have a sequence that varies from the reference genome. So where you see colour here, so where you're seeing the orange and the blue and the green and the red, those are positions, individual positions, where the reads which have aligned to the reference genome have a sequence variation from the reference genome itself. So they have a sequence which differs from the reference genome. So these are points of variation in our BAM file. So we can zoom in a bit and see those slightly more clearly. You can also see it as an overview of how many of your reads disagree with the reference. So the height of this bar here is showing you how many of the reads at this position disagree with the reference. So in all of the cases that we can see here, it is most, if not all, of the reads that align to those positions are disagreeing with the reference genome. The other thing that we can see in this window that might be of interest is this little eye here. This sort of purple eye that we can see here is indicating that in this particular read there is an insertion at this position relative to the reference genome. So this read contains an insertion relative to the reference genome. So that can be useful information as well. So you can see that kind of information at this level. We can zoom in still further and see these things in slightly more detail. So if we zoom in and click on the coverage track at any particular position, we get a little summary of what's going on at that position in the alignment. So here we can see we have 120 reads that are aligning at this position in the genome, position 2,320,453, and 120 of those reads, so all of them are a C. So there's a C at that position in all 120 reads that align at this position. You can see that the reference genome at this position is not a C. It's whatever that orange colour represents, which I think is G. Yes, it's G. So the reference genome at that position is a G, but all of our reads that align at this position, all of those reads contain a C at that position. So this is perhaps a candidate change in the genome that we've got sequencing data from versus this E. coli K12 genome. And then a few bases downstream, in the reference genome, we have what I think is an A. I should really know these colours by now. Yes, an A. So in the reference genome, this base here is an A, but in our sequencing data, 99% of the reads that align at this position are a G. There's one A and one T at that position, but everything else is a G. So we've got 139 reads aligning, 137 of them are Gs. Okay, so that is some useful information that's available to us just at a glance in IGV. And so this is relatively useful. We can this information that's in the alignment about mismatches compared to the reference genome, we can use in a slightly more robust and statistical way to try and find genuine places where our genome that we've sequenced varies from the reference genome, which is something that we'll take a look at, not this afternoon, but we will take a look at. Okay, so some other things about IGV, just quickly. If we right-click in any one track, we get a context menu for that track, which is actually really quite long. You can't see the bottom of this menu on my screen here, but this is popped up a long context menu. This particular track, I'm going to sort my alignments. No, I'm going to. What am I going to do? I want to view my alignments by pair. Actually, there's an option a bit further down. It's off the screen. Let me see if I can right-click a bit higher and see if I can see. No, it's still off the screen. It's actually the first option below these things that you can see here. So below quick consensus mode, we have an option which is view as pairs, which unfortunately I won't be able to get to appear on my screen. But view as pairs will connect these reads up in their appropriate pairs. To view this better, we're probably going to have to. We are going to have to a little bit because what this does is it takes a pair of reads and it joins them up on a single line. So it kind of spreads the data out a bit more. So we're just going to zoom out a little bit to see this. So now here on this top row here, we can see a pair of reads here. So we've got the left hand read here, which has a little arrow and is pointing towards the right hand pair here, which is again has a little arrow and is pointing towards its pair, and they're joined up with a little line. So this is illustrating this. These two reads are a pair of reads. So again, like you can click on the coverage track, you can click on the alignment track as well, and IGV will pop up some information about your alignment. So in this case here, because this is paired end data, IGV is popping up not just information about the read that I clicked on, but also about its pair. So I clicked on the left-hand read here, which we have all of the data for here. This is extracted from the BAM file. So this is displaying to us the information that's in the BAM file for this read. But it's also pulled up the equivalent information for its pair. So for the second in pair, or sorry, for the right-hand side read. In this case, actually, the left-hand one is the first in pair, and the second, the right-hand read is the second in pair. That isn't necessarily going to always be the case. But in this case, that is that is true. That the left-hand one is the first in pair, and the right-hand one is the second. Is in this summary that is pulled up by extracting the information from the BAM file. A lot of this data here is actually extracted from the bitwise flag that we've talked about relatively extensively now. The information about whether the mate is mapped, whether it's the first in pair, and those sorts of things, that's extracted from that bitwise flag. So IGV is presenting us with this data as it's pulled out of the BAM file. So we don't necessarily have to be able to read and understand those SAM files to be able to extract this data in a meaningful way, because IGV will do it for us. Okay, what else can we do? We can change the way that these alignments are coloured in. So at the moment, all of the reads are just coloured in grey, so they're shown to exist, but that's all the information that we get. We can get more information about the reads by colouring them in different ways. there's an option in this context menu when we right click the track to color alignments by, and I'm going to color the alignments by the first of pair strand. Okay, so this will color them either blue or kind of a pinky color as to whether the first in pair read aligns to the top strand or the bottom strand, so whether it's going right to left or left to right. And what we should find for this kind of genome sequencing data that we've got here is that whether the first in pair read aligns to the top strand or to the bottom strand should be pretty random, should be about 50-50. And it's difficult to get an overview of that with such a zoomed in view where we don't have very many reads in our window here, but you can see there are some of these are pink and some of them are blue, so you've got pairs that have different orientations within the same region of the reference genome. If you were viewing an RNA-Seq experiment in IGV, which if you've produced an alignment, which we didn't when we did our RNA-Seq analysis, we didn't produce an alignment. But if we had, using an RNA-Seq aligner like STAR, for example, if you view an RNA-Seq alignment in IGV and color it in this way by the first in pair strand, you can get an idea about the stranding of your experiment. So in an RNA-Seq experiment, all of the fragments which align to a particular gene should all align with the first in pair read on the same strand in a stranded experiment. So in an experiment where we've got a stranded RNA-Seq experiment, you should see blocks of the same color for reads that all align to the same gene. And if you see a mix of colors, so if you see pinks and blues within the same gene, that will usually tell you that that experiment was actually unstranded. That's not very common anymore, but it can be quite a useful thing to visualize. Okay, so you can color these alignments by lots of things. Not necessarily. I've done first in pair strand here, but there's all sorts of options in the color alignments by here. So we can color by the insert size, by the orientation of the pair, and various other things as well. So color it in by insert size, and it colors some in by looking at those, they look like they have quite big insert sizes, so they've been colored in by that. Let's put it back to first of pair strand. There we go. Just to draw attention to these, so here we've got some other positions where the reads are varying from the reference. In this case, it's not the case that every single read contains the varying base. So in some cases there are alternative bases at this position. So here, for example, in this particular position, the reference genome is a C. We can see that from the bottom, from the genome, the gene track down here. It tells you the sequence. So the reference genome is a C, but we have at this position only 69% of the reads that align here are C. So we've got 30% of our reads aligning here are Gs. So it isn't universally the same as the reference genome, but this is not as clear cut as some of the cases where we were seeing all of the bases being an alternative sequence. So in this case, actually most of the reads are still supporting the same base as the reference genome, but there's enough discrepancy for IGV here to highlight it and to show us that there is some there are some discrepancies at this position. So it does that by colouring in the coverage bar like this. So you've got it coloured in predominantly blue because most of the bases are blue. Then you get this little stacked bar chart view of that particular base. Okay, so this is a relatively kind of high level overview of IGV and some of the very basic features of IGV. It's much more complicated than this as a piece of software, but it's quite it would take a long time for me to go into a lot more features, and at this point, without having any real reason to illustrate those features, it doesn't seem I don't want to go into too much more detail, but hopefully you get an idea of the utility of it as a piece of software for visualizing, particularly in this case our alignments, and it is quite good as a tool for viewing alignments, even really quite large alignments. This alignment isn't huge because it's against bacteria, but you can load in whole human genome sequencing data where your BAM file can be up to anything up to 100 gigabytes or even more, and IGV with some persuasion will handle those very large BAM files, and that is because of the index. That's why it requires the index, because actually what it reads is the index and then uses the index to extract only the reads for the part of the genome which is visible. So that's how IGV can handle even very, very large BAM files, as long as you keep your kind of visualization window quite small. So in my case, I could zoom out to 500,000 bases, and it's still showing me the reads. Obviously, to be able to do this, IGV needs to load quite a lot of data into memory. You probably don't necessarily want to be doing this, and you can change in the settings. So if you go to View and then Preferences, this pops up the preferences, and you can change that zoom. So visibility range threshold is what you want to change. So at the moment, my visibility range threshold is 1,000 kilobases. If I change that to 100, my alignments here will disappear because I'm viewing near 900,000 bases. So I would have to zoom back in again to 100,000 bases or less to be able to actually see the alignments. So those kind of things are important especially when you're viewing very large files. Here we're working with a relatively small BAM file, and so it isn't too bad to visualize things in this way. Okay, so I think with that I'm gonna call it a day today. Thanks for tuning in, everybody. It was just as I say, fairly high-level overview of what I think is a very useful piece of software, and one which we will almost certainly be coming back to in future sessions when we some different types of alignment. Next time out, we're going to think a little bit more about these points of variation that we see in this genome. So in our alignment versus our reference genome, we are seeing places where the reads which we've aligned exhibit variation compared to the reference genome. And in session number 48, we're going to think about that variation and how we might kind of pin that variation down using some sort of statistical framework. This, again, is going to be fairly... I'm not going to be going into... So we're going to be doing variant calling, but I'm not going to be going into an awful lot of detail about kind of really nuts and bolts variant calling, especially using kind of human genomes and things like that at this point, because that's very, very involved. So we're going to be using BCF tools to call variation in this E. coli genome versus versus the K12 reference. So that's our task for next time around. Okay, so thanks for tuning in. Just quickly to have a look at the live comments. Dan seems to be having some trouble getting IGV installed. I would hope that for Mac and for Windows, it shouldn't be too bad. For Linux, I would install it via Conda, but I've not tried, not for a while anyway. So I can't really speak to that very well. And then Graham at the end asked, can you put UCSC tracks into IGV? The answer to that is absolutely yes. If something is available as a file format which IGV understands, then you can load it into IGV. And if you want to know about the file formats that IGV understands, then there's a file formats section on the IGV website here. So there's a list there of all of the kind of files that you can load in to IGV. You can download data out of the UCSC Genome browser by going to the table browser. So in the table browser, you can download data for all sorts of data from here. So here's gene and gene prediction tracks, but there's all sorts of information that's available from the UCSC Genome browser via the table browser. So you can download data out of UCSC from here in a supported format, so GTF, bed, all sorts of other things, and load it into IGV. So yes, if there's particular information that is available in UCSC that you want to see in IGV, and then you can download it out of UCSC using the table browser and load it into IGV. Okay, I'm going to call it a day. I will see you all next time out. In lockdown learning number 48, but that's the end of lockdown learning number 47. So I'll see you next time. So stay safe, keep well,